Hello, my name is Stephen Reppert. I'm a professor of neurobiology at the University of Massachusetts Medical School. And this is my second talk on the neurobiology of the spectacular migration of the Eastern North American monarch butterfly. And this second talk is going to be focused on a major compass system the butterflies use during their journey to arrive at Mexico, what's called a time-compensated sun compass. So we'll go through this in considerable detail. So this is a cartoon of North America and shows the major flightways in yellow of the Eastern North American monarch butterfly, starting off in the north and making this long journey down to Mexico. As I mentioned before, the butterflies are on their maiden voyage that undertake this journey. They're at least 2 generations removed from the butterflies that had migrated there the year before. So we're very interested in trying to understand the genetic program that's turned on in these butterflies that allows for this spectacular behavior, and particularly focused on navigation and orientation mechanisms. Now let me review again what I mentioned in my first talk about just a very brief overview in this next animation about this time-compensated sun compass, what it is. And it is a system whereby the circadian clock compensates for the movement of the sun, which is moving across the horizon over the course of the day, providing directional cues to the butterfly while allowing the butterfly to still maintain a southerly flight direction. So this cartoon shows how we're going to talk through the different components of this sun compass system. We're going to start first with the skylight cues that the butterflies use for directionality. Then we're going to discuss how this is sensed by the compound eye of the butterfly, how this information is then communicated to an actual structure in the center of the butterfly brain that functions as the sun compass. And then we're going to spend also a considerable amount of time talking about the circadian clock and how these two systems, we believe, crosstalk with one another to lead to this sun compass system. First, let me tell you about how we monitor behavior in the butterflies, because you have to have a behavioral readout in order to do experiments. And what we use is a flight simulator. A cartoon of that simulator is shown here. It's modified from one developed by others, but this is our iteration of it. It's essentially a white opaque barrel that's open at the top. We fly the butterflies outdoors in Massachusetts, and when the butterflies, which are positioned in the center of the barrel, can only see through the top of the barrel the blue sky and the sun. Now the butterflies are tethered so that they can be positioned properly in the center of the barrel. They're attached to a guide wire that actually goes up to a strap across the middle of the top of the barrel. And by this system, it's wired in such a way that the output is to a very simple computer program where we can track the actual orientation of the butterfly in the flight simulator. So the butterfly is fixed and can only rotate horizontally, so it can move in all 360 degrees of directional movement. And that can be recorded again very precisely by computer. Now the second thing that's essential about this system is to have a video display, or a video camera, I'm sorry, hidden in the bottom of the barrel. So really the experimenter who's in a remote location has a computer screen to see the readout of what the actual orientation of the butterfly is. But you have to have continual video observation that the butterfly is actually flying to use that computer-generated orientation data as an actual data point. So the rule is we will have butterflies that, for example, fly continuously for 5 to 10 minutes before we can use those data then as an actual data point for an experiment. Now, let me give you, I guess, a bird's eye view of butterfly in the flight simulator, a very short movie clip to show you how directionally these butterflies, comfortably, these tethered butterflies fly. So you can see this, the butterfly is flying really very directionally to the southwest. I move the butterfly out of the way, and you see what happens? That butterfly wants to go right back to that same orientation position. So this just shows us and you how robust this behavior is and how well it's manifested in the flight simulator. And this and other control experiments give us great confidence that what we're looking at in the flight simulator has, we believe, some basis in the reality of butterflies actually migrating outdoors in the real world. So let me take you through a simple experiment. And this experiment will accomplish two things. It will show you how we actually deal with the orientation data, how we plot them and analyze them. It's very simple. And in doing that, I will take you through an experiment that is also pretty straightforward that will demonstrate the critical importance of the circadian clock for this sun compass activity. So in the first part of this experiment, we had butterflies that were captured in Massachusetts, and they were housed in a light-dark cycle that mimicked what it was at that time of year, with lights on at 7 in the morning, lights on at 7 in the evening, off at 7 in the evening. The butterflies were housed in this lighting condition, again simulating outdoor conditions, for a week until they were brought outdoors to do flight simulator studies. We then flew those butterflies at 10 o'clock real time, which is also 10 o'clock butterfly time, because the butterfly circadian clock is coordinated by the prevailing lighting conditions. So here are the results shown on the circle diagram. And this large circle represents all 360 degrees of orientation position. By convention, north is on top, magnetic north. And then the small blue dots represent the mean flight path of an individual butterfly. So what we're looking at is a group of butterflies, 13, who flew continuously for 10 minutes, were directional, each individual, and then one does circular statistics on the group of animals, and you can see by the length of the arrow that these animals were highly oriented to the southwest direction, which is what they need to, where they need to be oriented to make it from Massachusetts to Mexico. That's the position of the sun relative to the circle diagram of direction. Second part of the experiment. What we wanted to do was very simply, and this is because of the great directed behavior of the butterfly over the course of the day, allows us to do this sort of study. What we wanted to do is to shift the circadian time of the butterfly, put it out of sync with environmental time, and see what happened to the orientation direction of those butterflies. So what we did is we put butterflies in the lab. This time, these migrants were exposed to a light-dark cycle that was advanced by 6 hours. Lights went on at 1 o'clock in the morning, off at 1 o'clock in the afternoon. We flew the butterflies again at the same time we flew the first group, 10 o'clock. But this would actually be, according to the butterfly, that sun position would be interpreted by the butterfly as 4 o'clock because its circadian clock has been shifted. So if it really views this as a 4 o'clock in the afternoon sun, then the butterfly should predictably shift their orientation in a counterclockwise direction so that they're now flying so that the sun is to their right as they're flying, they think to the south, but actually to the southeast. And that's exactly what happens. So these butterflies now have shifted their orientation. Instead of flying at 10 o'clock in the morning, the sun to their left, they're now interpreting that sun as a 4 p.m. sun. It's to their right as they're flying to the southeast based on what I just showed you. So this study shows how important the circadian clock is, and I will show you other studies that we have done where we have eliminated the circadian clocks that are important for this sun compass system, and those butterflies as a group are completely disoriented and would never make it to Mexico and be able to fulfill their biological destiny. Again, emphasizing the importance of the circadian clock. Now, what are the skylight cues the butterflies use for directionality? Well, this is a cartoon, a very standard cartoon one shows of an observer, which in this case is the monarch butterfly, with its sort of dorsal thorax pointed towards the top of the sky or the zenith, the overlying celestial hemisphere. And this butterfly is looking in this position at the sun. And it's important to realize that the sun, its azimuthal or horizontal position, is the dominant skylight cue for directionality that the sun compass uses. Now we see as emanating out from the sun in these sort of circles that are sort of radiating out from the sun, which are made up of these bidirectional arrows or so-called electric vectors. This is the polarized skylight pattern that is a result of sun, of the rays of the sun being scattered, from the sun being scattered off particles in the atmosphere at more or less right angles, giving rise to a polarized skylight pattern that moves as the sun moves. And so animals that have the right kind of eyes, which the butterfly does, can exploit this so that the monarchs on partially cloudy days, when the sun is obscured, as long as they can see a patch of blue sky, can continue their southerly flight direction. So this is our working model of the clock compass system. And again, I'm going to continue to focus on taking that skylight information and talking about how it ultimately gets to the sun compass. So the skylight information is processed by the compound eye of the monarch butterfly. And the retina of the butterfly is compartmentalized into two main regions for sensing these two different cues. First, there's the so-called dorsal rim area, which monarchs have and virtually all insects have, which is an area of the eye which is anatomically and molecularly specialized to sense the angle of monochromatic plane polarized light. And in fact, we and others have shown that this dorsal rim area in the butterfly is only responsive to ultraviolet polarized light. All the photoreceptors in those ommatidial units only express the UV opsin. Electrophysiological studies of the dorsal rim show it only responds to ultraviolet polarized light. And at a behavioral level, we've put a linear polarizer above butterflies in a flight simulator and rotated it, and have also shown that at the behavioral level, the butterflies can respond to changes in the angle of polarized light as long as it's of the UV spectrum. The main retina is where all the ommatidial units express the three basal opsins, UV, blue, and green, in a particular array, which allows for color vision. And the main retina is also where the horizontal position of the sun is sensed. So once this information is sensed by the eye, it's communicated by a complex circuitry that we're just beginning to get a handle on, and ultimately ends up in the central brain region, which is designated CC, which stands for central complex. And that is, we believe, the site of the sun compass, integrating sensory information about these skylight cues, and also sensory information from a number of other modalities. But in this context, really the important feature is integrating information about skylight cues and using this central complex as an actual sun compass system. And then the circadian clock interphases downstream of that processed information, skylight information, ultimately affecting the motor system, and then leading to oriented flight behavior. Now, this is a very striking image of the monarch and butterfly brain. It's a pseudo-colored image, and it's a result of synapsin staining, confocal microscopy, and three-dimensional reconstruction. It's a frontal view, and the pseudo-colors are so I can point out just the main structures of the butterfly brain. This is a frontal view, and really let you know where the central complex region is anatomically that we believe is the site of the sun compass. So you have the optic lobes, which are huge because this is a day-active animal, and we can see that they're colored in yellow. And then we have the central brain itself, where, for example, the mushroom body lobes are shown bilaterally symmetrical in red. But the structure that we really want to focus on is this midline structure, again, CC for central complex, which we believe is the actual site of the sun compass. Now this anatomical work was done by a postdoc in the lab, Stanley Hines, who has moved on, is now at the University of Lund, or Lund University, I should say. And Stanley not only did just the overall anatomy of the monarch butterfly brain, but he really focused in to get a higher resolution image of the central complex and related sun compass structures. So he actually generated a standardized sun compass neuropil. And so what is shown here is a three-dimensional reconstruction of the brain that is now grayscale, except for the what is colored are the structures that are felt to make up the central complex, the sun compass itself. Not only the central complex, but related structures. So collectively, this would be the sun compass system. So this higher resolution standardized sun compass structure I'm going to extract this out of the brain so you can see it now in a better view. We have the bilaterally symmetrical anterior optic tubercles shown here. These are major input pathways to the hub of the central processing, the central complex itself, processing sensory information, particularly skylight cues in this instance, and then output pathways through these lateral accessory lobes on either side of the central complex. Now, having this standardized sun compass neuropil allowed us to do volumetric studies, for example, look at changes in these various regions over the course of the migration. But the one thing that Stanley did next, which I thought was most important, was to put down sharp electrodes into this sun compass structure, inject dye into single neurons, and then reconstruct them and register them on this standardized sun compass structure. And what Stanley was able to do was to actually identify over 30 different cell types in the central complex region. Here is a composite of all of these registered neurons, again registered on this standardized sun compass structure. It's a very dramatic image of these cells and their arborization patterns. So we have a hardwire map, if you will, of the major input cells to the sun compass, intrinsic cells and their arborizations, and output cells. But this is just wiring. And so what we need to do now, since we have this, which is great, is to make these functional, to look at functional circuits based on what we've begun to do with this wiring diagram. So one of the first things that Stanley did was to put down a sharp electrode into the sun compass structure, and these initial studies were aimed at cells in the input stage of this system. So these were done in the laboratory where a butterfly was immobilized, electrode was placed into a neuron. Once a stable firing rate was captured, then in the laboratory, Stanley could rotate the major lighting conditions in a very controlled way and see how that single neuron responded to changes in the skylight cues. So we could rotate above a linear polarizer that would rotate the angle of ultraviolet polarized light, and then it could rotate around the butterfly in all 360 degrees of direction, light spots that would mimic azimuthal position of the sun. And what he showed remarkably was that these individual neurons were actually responding, each neuron, to both the azimuthal position of the sun, but also to the electric vector angle. So these two sources of skylight information originating from different parts of the butterfly eye were already being integrated in these single neurons. And this dual integration seemed to provide a consistent representation of skylight cues in the sun compass over the course of the day. Therefore, we could then increase our understanding of how the butterfly on a cloudy day is able to pick out a patch of blue sky at any time over the course of the day and use that information to continue to maintain the appropriate directionality. This is just the start. With this information, now our work is really cut out. And so what we're trying to do is more physiological recordings, not only single cell recordings like Stanley did in different regions of the sun compass system, but also in multi-unit recordings, and particularly at the output end of things, because ultimately we want to understand where the circadian clock is compensating this integrated skylight information, because that's really the million-dollar question for us. The other thing we're doing, we have a genome. We have used nuclease techniques to knock genes out in the butterfly. Therefore, we want to use that to knock reporter genes in so that we can do this sort of circuit mapping in the butterfly that is now commonly done in worms and flies and model organisms as such. Okay, now let's move to the clock component of the system. And there are two regions in the head that we have focused on circadian clocks. First, talk about brain clocks. And there are 3 cells on either side of the brain in this so-called pars lateralis region that we have strong evidence are, in fact, the location of the major circadian clocks in the butterfly brain. Now, we have terrific antibodies, highly specific for the monarch clock proteins to stain for their existence in tissue. I should mention we have a monarch cell line that has a light-driven clock. So we can use that cell line, which expresses all the clock proteins, to verify, for example, our antibodies, knock down those proteins and their antigenicity, and in essence, validate the specificity of each of our antibodies, which we have done. We've done other work to really show that these cells in the pars lateralis, we believe, are clock neurons. So what do brain clocks do in butterflies? They do similar things to what they do in us: regulate sleep-wake cycle, regulate hormone rhythms, metabolic rhythms, but they also do things we think special to the migration syndrome of the butterfly. Now, in terms of the brain clocks, we've identified a few years back a pathway from the pars lateralis to this structure, which is the pars intercerebralis, the so-called hypothalamus of the insect brain. So, so we have identified a neuronal pathway that connects these 2 regions. So what we believe is happening is that the circadian clocks are tracking the decrease in day length that occurs in the fall, in giving rise to migratory butterflies eventually. And that information is communicated along with decreasing temperature to the pars intercerebralis. And then downstream, this system is activated, so there's actually a decrease in insulin-like peptides, leading, in essence, to juvenile hormone deficiency. Juvenile hormone deficiency in migratory butterflies has been known for decades. And that is what is responsible for the decreased reproduction and increased longevity in migratory butterflies. But we now are beginning to get a neurobiological substrate for how this decreased juvenile hormone production actually can occur in migratory butterflies. So what about the time compensation of the circadian clock? I'm sorry, of the sun compass. Time compensation of the sun compass. Where are those clocks? One would expect they're in the brain. Makes perfect sense, but they're not. Those clocks are actually in the antenna. So the antenna contains circadian clocks whose molecular control, which I'll go through in a few minutes, is identical to what has been found in brain clocks. And those antennal clocks are both cell and tissue autonomous, so that you can take the antenna out and those clocks will respond to light independent of being connected to the head. So you have antennal clocks that can actually act independently from brain. Let me just tell you how the first experiment we did, which was a very simple experiment, not to really take away from many more sophisticated experiments that we've done. And these initial experiments were done by Christine Merlin when she was a postdoc in my lab, who is now at Texas A&M. At least she spearheaded these studies, which involved a number of people in the lab. The first thing was just what happens when you remove the antenna from butterflies. And here are the circle diagrams again, and what you find in intact animals. These animals, again, are highly oriented to the southwest. And what was found with antennaless butterflies is that these butterflies, as an individual, are directional. They continue to maintain a fixed direction, but as a group, they are completely desynchronized one from the other. So we believe what this is telling us is that in these antennaless butterflies, without the time compensation component, the sun compass system has already been integrated, so these butterflies will pick a direction and continue to go, but it's not time compensated. So if you look at a population of butterflies, they're all over the map. We've done further studies. These were done largely by a postdoc in the lab, Patrick Guerra. And what Patrick showed, which is I'll just highlight in cartoon fashion here, is that you only need one antenna, either the right or the left, sufficient for orientation behavior. And he's shown using that and molecular approaches that the two antennae do not talk to each other. So there's no crosstalk between the antennae. And he's also shown that if you take one antenna, for example, and paint it black, which essentially blinds that antenna and desynchronizes all the clocks so they no longer know what time it is, they're running out of sync with each other. And you paint the other antenna in the same animal clear, and we know that clear painting the antenna has no effect on orientation behavior. Now, putting these two antennae in conflict with each other, those butterflies are completely desynchronized in terms of their oriented flight behavior. So we believe that the information from each of the two antennae is actually incorporated or is integrated at an integration site in the brain, and then this information ultimately communicated to the sun compass system. How we get from the antenna to the sun compass structure, the central complex, we have some ideas in terms of circuits that may be involved, but we're really at the beginning stages of figuring this out. Now, what about the molecular clock mechanism in the butterfly? It's also really pretty interesting. Before I talk about the molecular clock in the monarch, let me just review very briefly what we know about the main gear of the molecular clock of the two most well-studied animal models, Drosophila and the mouse. And this will provide an important background for the studies I'm going to be talking about in the monarch butterfly in terms of its clock mechanism. So here's Drosophila. Again, these are highly schematicized models and has this standard negative transcriptional feedback loop, which is intracellular, which you're probably all familiar with. And basically you have two transcription factors that are driving the regulation of two clock genes per and timeless. And as these proteins are translated, they form complexes, and 24 hours later, they go back into the nucleus to shut down transcription. Now, the other protein that I want to really emphasize is what's called cry, which stands for cryptochrome. It is in the same clock cells in the fly, but it is not an essential part of this negative feedback loop. It's external to it. And cry stands for actually cryptochrome. And this just shows the crude chemical crystalline structure of cry from Brian Crane's group at Cornell, reported a few years back from Drosophila cry. And I just show it to remind me to tell you that this black portion of this molecule is the binding pocket for the chromophore flavin. So cryptochromes are flavoproteins, and that's what allows them to actually respond to light. And in the case of Drosophila, its cryptochrome functions as a blue light circadian photoreceptor. So on light activation, cry interacts with timeless, causes its degradation, and resets the molecular clock. And this occurs at dawn and dusk, and in this way, the clock is reset to the 24-hour day. Now let's look at the mouse model. And what we see is a negative feedback loop that's very similar to what is found in the fly. But in the mouse, we have families of genes that are being regulated because we've gone up in terms of the evolutionary scale. So we have transcription factors similar to those found in Drosophila, regulating now two families of clock genes. We have the period gene, 3 period genes, and 2 cryptochrome genes. And now in the mouse, it's the purrs and the cries that form complexes, and 24 hours later go back into the nucleus, where it's cryptochrome that's the major transcriptional repressor. So a lot of similarities between these two clocks in the fly and the mouse, but there are differences. And the one difference I want to point out is this potential function of cryptochrome. Here it's a blue light photoreceptor. Here it is part of the molecular clockwork itself. And this function in the mouse, importantly, is light independent. Now, when these two models were proposed around 10 years ago, what we all thought was that over the course of evolution, the Drosophila cry had undergone a gene duplication event as to what we find now in the mouse, and the molecule had been modified somewhat and intercalated into the clock. That's not correct because of what we found in the monarch butterfly. In the monarch butterfly, we found one of each of the two versions of cryptochrome based on an EST database. These were studies first reported in around 2006. And basically, the butterfly has what's called type 1 Drosophila-like cry, which I'm calling cry 1, and will be designated in orange. And the important thing is that the butterfly has the second cryptochrome, which is the type 2 cry, vertebrate-like cry, which we call cry 2, which will be shown in green. And in cell culture systems, again, in the cell line that we have, this monarch cell line, where we can construct the circadian clock, we've shown that the Drosophila-like cry functions as a blue light photoreceptor in the butterfly like it does in the fruit fly, and that cryptochrome functions as a major transcriptional repressor in the butterfly like it does in the mouse. So based on a huge amount of studies and reconstructing the cell line, then going to the butterfly brain itself, we now have this model, which is similar transcription factors to what occur in the fly and the mouse, regulating 3 clock genes per timeless in the vertebrate type cry. The proteins are made, they form a complex. 24 hours later, back into the nucleus, and it is cryptochrome, like in the mouse, like in our clock, that's the major transcriptional repressor in the butterfly. So the negative feedback loop is more mammalian-like than Drosophila-like. So this was a novel clockwork mechanism at the time. We started looking at databases, and what we found was really interesting, that none of the Drosophila species, all of them that I know of have been sequenced, have the type 2 cry. None of them. But all other insect genomes that are in the database contain the vertebrate type cry. So this does not mean that Drosophila has led us down the wrong path. Drosophila has been a tremendous animal for, and its genetics for allowing us to understand about how the clock works at the most fundamental level, and has guided a lot of the mammalian work that came after. Now, since we have shown that cryptochromes, we have these 2 types of cryptochromes, there are 3 ways we've proposed in which insect clocks can be put together at the molecular level based on these 2 cries. There's the ancestral model, which is shown here, typified by the butterfly, where I've already gone through, you have 2 cries. Then there's the derived clock in Drosophila, where the vertebrate type cry has been lost, and in the circadian clock cells, the light-sensitive cry of Drosophila functions as a blue light photoreceptor. So that's 2. Then there's a third possibility, which has been found many times now, and that is that there are insects which have lost the light-sensitive cry and only have the vertebrate type cry. That includes beetles and all hymenopterid genomes that has so far been examined. So their clocks, and we've done molecular studies in culture showing that this acts as a repressor, so that these clocks in the beetle and hymenopterans are even more vertebrate-like. So sort of take-home message, I guess, is that if you're studying non-drosophilid insects, there's no such thing as a non-drosophilid insect having a Drosophila-like clock. It can't, has to be different, because all other insects have the vertebrate type 2 cry. And these all emerged out of our initial studies in the monarch butterfly. So to get back to our journey south, and just to summarize the main points that I've tried to get across, that really the major compass system for the butterflies is this time-compensated sun compass used for directionality during their long journey south. That there is an actual sun compass, it's in an actual region of the brain, the primary component being the central complex, where skylight cues are integrated. That circadian clocks for time compensation are not in the brain as we had expected originally, they're in the antenna. And that the monarch molecular clockwork is cryptochrome-centric, which has led to the discovery of vertebrate-like cries in all these non-drosophilid insects that I just mentioned. So let me just end up by saying that there's one other feature of the monarch migration I just want to mention about orientation cue hierarchy. And it should be, I think, self-evident from what I've said The sun compass is dominant, with the sun being the primary stimulus on sunny days, position of the sun, polarized light on partly cloudy days. But it's been reported for decades that butterflies on completely overcast days continue to migrate south. So it's been postulated that they may have a magnetic compass, and a recent study has provided, I think, really clear evidence that they in fact do. But and so that this would be a system that is used as a backup system on completely overcast days. But these studies are the topic for another day. I want to end by showing this evocative drawing that Vivian Budnick did of the monarch butterfly migration. I want to acknowledge the funding agencies that supported this work, NIH and the Air Force Office of Scientific Research, and particularly all the colleagues in my lab who've been involved in these studies, as well as colleagues in other labs in neurobiology at UMass, and also colleagues outside of UMass. So that's the end of my second talk. And again, I thank you for your time and attention.